All right, welcome everyone. My name is Gina O'Connor. I'm a grant specialist with the Office of Research and Sponsored Programs. Due to USD's recent Deb Score award, we wanted to use this opportunity to educate faculty on how they could potentially get involved with funding from the DoD, as the DoD and its branches are committed to funding basic research and emerging research areas in the natural and social sciences, among others. Today, towards the end of the presentation, to ask a question, you can utilize the Q&A function at the bottom of your screen, or you can simply drop your question in the chat. Or if you want to raise your hand, I can unmute you as well. So it's really up to you. Today, we're excited to be hearing from Dr. Gregory Bertsch. Dr. Bertsch earned a PhD in biomedical engineering from USD and has been a faculty member in the Department of Entrepreneurship, Management, and Marketing with the Beacom School of Business since July of 2018. Last year, his team published an article describing work that has been done to create a wound dressing that fights infections. He's also working on commercializing paints and coatings that kill bacteria and viruses on contact. Today, we'll hear more about his research and experience in working with the US Army and the DoD. So with that, I'll invite Dr. Bertsch to get started.
Great. Thank you, Gina. I appreciate that introduction. I assume everybody can hear me okay, right? I want to do two things today. As Gina mentioned, yes, I've worked with the Department of Defense, I guess really since 2017. And so I want to talk a little bit about the research that we did, particularly with relating to the wound care technology. But I also want to, in the spirit of what we're talking about here, share a little bit about, what I've learned in our experiences in dealing with particularly the US Army. And so that's my objective. And if I can get the slide to change, there we go. And so you're going to hear over the course of these brown bag sessions, different people talk about different ways that they've interacted through grants, funding, et cetera. Our strategy may be a little bit different. We decided to engage with the Department of Defense as a small business, and particularly as a spinoff from a university or startup. And we did this because we knew that the Army and the DoD in general has really taken on an initiative to engage with small businesses in a more meaningful way. So we leveraged that and decided really to pursue that approach. And what you see here, there's a link that I've also included. This is an example of the focused effort and the specific organization that they've established really to work with small businesses. So there's a complete team right now that's trying to enhance and improve that engagement. So if you're interested, check out that website. You'll learn a lot. There are a few different programs that are more research focused. Some of the small business engagement initiatives are more general, maybe for existing companies. But from a research perspective, they do have some options. One is an option that they call SBIR, STTR. That stands for Small Business Innovation Research. STTR is Small Business Technology Transfer. It doesn't fit, I know, but those are more of like a traditional grant solicitation, proposal, grant kind of approach. There's also a program called XTech. And XTech is actually more competition-based, and we've done both actually, and we've had some success in both. XTech is kind of like pitching like a Shark Tank sort of environment, and I'll talk a little bit more about that later. But it's different. It's not quite your proposal-based sort of research funding. But starting with SBIR. SBIR is again very much like they submit a solicitation, you respond to that solicitation with a proposal, and then you're given a funding award. They actually call it a contract. And generally, this will focus on some kind of commercialization of the technology. It's a two-phase program. There's a phase one that's in the $175,000 range for 6 months. And then if you're successful, there's a phase two, which is in the range of 1.5 million, 24 months, and some potential follow-on funding, depending on, again, your success and Army interest. But this SBIR program actually is across all branches of the DoD. So in our case, we did US Army, but just about every group, like the Defense Health Agency has an SBIR solicitation, DARPA, the Defense Logistics Agency, etc. And this could be a pretty interesting option, again, for particularly university spinoffs. Success rate is around 15% for the phase one and 40% for phase two. And we've gotten a handful of these awards now. Again, it's been successful for us. The other program I talked about is this XTechSearch program. And again, this is more pitch-based, it's a competition. And it starts off initially pretty straightforward application. If you have an innovation or an invention that you think the Army might be interested in, you can write a white paper. It's generally maybe a couple pages long, and if that's accepted, you go to the next stage and the next stage and so on. And as you progress through these stages, you go from white paper to actually in-person pitches, and eventually you can win up to $250,000 as a finalist, which we actually were lucky enough to be a finalist in one of the first, I think it was the second round of XTech competitions back, again, like 2019 timeframe. So that's another way to leverage the small business angle to get research funded by the DoD. We've learned quite a bit along the way, and I just wanted to point out some specific lessons that we have taken away from, and that we are very diligent about including in our future proposals. One is really developing an in-depth understanding of the problem that the DoD wants to solve. This is really, really critical. If you can align yourself with a problem that's strategic, you're going to have much more success. And I know that's critical in all proposals for those of you who are doing research, but the DoD is extra critical, is what we found out. The other thing the DoD really likes to see is some sort of a market application in the civilian market. The DoD doesn't want to be your only customer in most cases. There are, of course, some sensitive technologies maybe where the DoD wants to own it all, but for the most part, they're going to want to see some sort of a capability for you to find customers outside of the Department of Defense. The last thing that I wanted to talk about was just the complexity of the Department of Defense. And this is the organization that we dealt with, the Medical Research and Development Command, or MRDC. And you can see there's 10 different agencies and groups underneath the MRDC. And each of these groups is hundreds, if not thousands of people. And so trying to navigate this whole ecosystem can be challenging. And so it took us a while to kind of figure that out. We work primarily with the Institute of Surgical Research. They're focused more on wound care and also burns. But we also interacted a lot with Walter Reed and also the Medical Research Institute of Infectious Disease to a lesser extent. But again, all of these people have kind of their own mission. They have their own facilities and labs and their own experts. So that can be challenging. All right. Good. I want to switch gears now and talk a little bit about our specific project. Or this specific project with the US Army. I hope the other content and information was helpful. But we we started working with the Army. Again, our first interaction on this particular technology with related to wound care was through the XTech competition. And we were able to be awarded an award as a finalist of $100,000. And so we leveraged that money to start research, and applied research, I guess is probably more correct, on this particular technology. And a little bit about our invention. So we leveraged a platform technology that we had called an enhalamine, which is an antimicrobial molecule. And we combined that with some other features, such as some anti-adhesion properties, and also some materials, more advanced materials for wound dressing. And we proposed that to the Army, and that's the basis of where we started. The reason why we came up with the product we did was, again, digging into this in-depth understanding of the problem we solved. We understood, after doing a little bit of research, that the Army looks at infection as a hair on fire problem. It's 1 out of every 4 battle-related injuries is prone to get an infection of some type, and oftentimes those infections contain drug-resistant organisms that cannot be killed with current antibiotics. In burn victims, that number goes up to 1 in 2. And then another thing that was maybe timely for us is the Army now, fairly recently, developed a series of modernization priorities. And one initiative that's part of that modernization priority is the idea of prolonged field care. And what they recognize is in future battles, there is not a guarantee.
Could you try again?
There is not a guarantee of having air superiority. And so that means that the goal of evacuating casualties within 1 hour may not be sustainable or achievable. So prolonged field care, that initiative assumes that care may be needed in the field for up to 72 hours. So products that enforce that, in other words, prevent infection for a period of time while or before help can arrive, are very important. And so that's really where our technology fit in nicely. The other advantage that we saw is the ability to not just be an additional piece of content in a 50-pound bag for a medic, but we could replace existing gauze. So there really was no addition there. So again, that's kind of the thinking that we went through when designing our product. And so the solution that we came up with was a gauze that prevented biofilms, also killed drug-resistant organisms, incorporated a hydrogel product, which is an advanced wound treatment or wound dressing product. We also added hemostatic functions, which means it aided in stopping hemorrhaging. And again, it also replaced existing gauze. So that was the foundational product that we posed in our research. And the research focus on this project, again, it was funded as part of XTechSearch, but then we also got follow-on funding through the SBIR program. The focus or objective was on proof of concept. And proof of concept meant, in this case, number one, we were going to demonstrate that the primary product functioned correctly. In other words, some of the antimicrobial infection-fighting elements worked, but also it needed to demonstrate safety. It had to still be useful as a conventional gauze, and it had to be stable over some lifetime on storage on the shelf, etc. So that was essentially the objectives. Like I said, I wanted to just quickly talk a little bit about the chemistry. I put a link here to our paper that we published on this if you want more detail. But our solution really, our technical solution, involved a copolymer of three primary components. The first component was our N-halamine antimicrobial, which utilizes chlorine as an active ingredient, so it's very effective. And then we also had a portion of the polymer, copolymer, that used a product called DMA, which is a known adhesion promoter. And that allowed us to bind to different substrates, like our conventional gauze base. And then finally, we used an anti-adhesion component called the zwitterion that would repel biomolecules, which is also very useful in preventing biofilm formation, which I'll show a little bit more on here in a second. And so our job in this case was to improve the formula to a certain degree, but in a large degree, it was to demonstrate this proof of concept evidence. So one of the things that we did is we did a test on the antimicrobial efficacy, and you can see the results here of that test. Primarily, the test exposes some samples that we create to various types of organisms. This particular panel is known as the escape panel, and it's the group of organisms that are the most problematic for the US Army. And what we're showing here from a results standpoint is that we're achieving, and in this data, larger numbers are better. And so in a 6 log reduction at the top of that chart, you're looking at 99.999% kill rate. So the punchline here is that our technology is able to kill virtually all of these bacteria within 5 minutes, if not 5 minutes, certainly within 15, which is not surprising. That's really kind of a fundamental benefit of using N-halamines. But the other thing we needed to do is show our ability to resist adhesion for biomolecules. So on the right, or I'm sorry, on the left-hand side, we used a protein, it's called fibrinogen, which was tagged with a fluorescent molecule. And we coated certain surfaces and then rinsed them, and then we examined them under a fluorescence microscopy. And so ones that glow have a lot of proteins attached. The ones that are black have little or none. And so you can see some differences. There are a couple of different variations or versions that we tested. But generally, we're much better than conventional gauze. We also compared it to a commercially available non-stick gauze, and we had similar performance, I would say, to that product, which was encouraging. On the right, we tested actually the safety or cytotoxicity of our product. So we took a sample and we exposed human skin cells to our sample plus some controls. And the results here indicate that a chlorinated HaloCare, which is our brand name of this product, did not perform at 100% cell viability. So there was some toxicity in the product, which wasn't a huge surprise. But one of the things that I think was interesting to learn is that we did much better to a silver infused gauze that's commercially available. So our takeaway here is that we are significantly safer than some antimicrobial products currently on the market that are used and proven to be somewhat effective. So that was encouraging as well. Another feature that we needed to demonstrate was that our product would not hinder the performance of the base gauze that it was intended to replace. And so these are some standard material property tests that are typically done on textiles. I'm not going to read through each one of these, but we're looking to make sure that the strength, the air permeability, and the stiffness really aren't affected. In general, it was very successful. The one thing that we did find is that our product was a little bit stiffer. Adding that polymer to the matrix did stiffen up the base product somewhat. And that's something that we would need to work on with some formulation optimization in the future. But that was one disadvantage that we did see, which is shown in the kind of the upper right-hand chart. You can see kind of the difference there in rigidity. And then lastly, we tested two things. One is storage stability. And for storage stability, we looked at the chlorine content. We measured that using iodometric titration. We checked that every 5 days over the course of 120 days. And we demonstrated that the chlorine was not leaving the product, that it was stable. And so at least for about a 4-month period, we felt that our material would still be effective. The last test that I've got listed here is the hemostasis characteristics. We added a hemostatic agent, which is a blood clotting agent, to the product, and we wanted to make sure that our science, our technology, didn't have an impact on that particular chemistry. And so you can see HaloCare performed very comparably to something called QuickClot, which is a commercially available product in blood clotting. And in this case, lower number is better, using something called the blood clotting index, or BCI. And so some takeaways here, next steps. First of all, moving forward, what we need to do, we've proven the concept, but we need to explore more in optimizing our product, some of the material property issues that we saw. Also, maybe from a cost perspective and efficacy perspective, there's probably some work we can do. And then some of these next points really connect to the push towards some regulatory approval activities. One being animal trials. One thing in wound dressing that's a little bit of a mystery is that you can have a product that performs really well on the bench, but then when you put it in animal trials, it's a complete failure. So we have to confirm that ourselves. And then there are some regulatory biocompatibility trials that are necessary that need to be completed. From a funding standpoint, what we intend to do is really solicit the USAISR, which is the Surgical Institute Research Center of the Army, and explore not just some of the advantages we see, but also leveraging this against their burn initiatives and through their burn center team. And so that's a next step in terms of looking at proposals. And so hopefully that was interesting. I guess I have Gina, about looks like 7 minutes left for questions, if there are any, that I'd be happy to answer. So thank you.
Yeah, thank you so much for your time. As a reminder to our audience, if you want to use the Q&A function, that works, or the chat, or you can raise your hand and I'll unmute you. But to kick things off here, you described the DoD as a somewhat complicated ecosystem. What was your initial first steps with your involvement with US Army XTech and SBIR? Was it information you got off a website, or was it someone that you met personally?
Yeah, good question. Actually, it was one of my colleagues sent me an email. He had seen it, and so there are ways that you can sign up for newsletters and various things, and a lot of the researchers probably are doing that already. I get 5 emails a day from the National Science Foundation, and he had happened to catch one actually from the Army, and it was what was striking about it is it was introducing this brand new XTech program. And so they were really pushing for applicants. So he sent that to me. We had some discussions, and then decided to enter. I never thought we'd get very far. And again, I think there were 400 people that entered, and by some stroke of luck, we ended up being a finalist. So it was a really fun. That's another thing. XTech was really fun, and quite an experience. So yeah, I encourage people to give it a try.
And now that you've been in it for a while, what specific reporting requirements does the DoD expect from small businesses involved in their projects?
Yeah, we had to submit a monthly report and then a final report. And one of the things that's different about the Army in particular, I can't speak for the entirety of the DoD, but the Army has very specific forms that you have to complete and very specific website locations where you submit different things. And there are different kinds of reports. The financial report is a little different than your progress report. And so navigating that can be a little bit tricky. You've got to read, and usually when you get an award letter, It's got tons of detail in it. So you have to be disciplined to sit down and read that and understand all of the different requirements, which speaks to a lot of the different resources that you have to engage with when you're submitting these reports. So that was a little tricky, but not egregious. I mean, not much different, at least than like a USDA grant or NSF grant.
Okay. Yeah, that sounds pretty average from what I know. Are there any restrictions on the types of businesses or entities that can apply for this SBIR program?
There are a few. You have to be under 500 people. That's their definition of a small business. On the flip side, they don't necessarily care if you just started, if you just formed your business today and you have one person. That's really kind of at the heart of what SBIR in particular is about. So that works out well for a lot of university researchers in particular. There are some requirements on citizenship and who could be the PI. So you might want to look at that. The PI also has some requirements in terms of their load. But again, like you mentioned, Gina, I think there are requirements like this for a lot of other regulatory or a lot of other agencies. So similar.
What advice do you have for other researchers on campus who might be interested in seeking some DoD funding?
Well, I guess it would be interesting if they have some technology. I think I would, offer to have a conversation with them to see where they might fit. It's just, it's so complicated, and well, there's so many options that it's hard to say, here's some advice that's going to work. I think this worked for us, taking this small business angle, but there are tremendous, there's a huge number of solicitations out there for different research projects. So trying to be specialized maybe might be my advice, as focused as possible on the problem that they're trying to solve.
But it sounds like from the small business perspective, your experience with working with the DoD has been rather good. Is that true?
Yeah, it has been. I've learned a ton. I've connected with a lot of really interesting researchers and collaborators. It's really been eye-opening from a lot of perspectives.
Yeah, exciting stuff. This was all news to me too. Are there any questions from the audience that anyone has? I'm just kind of scanning down here to see. Here's Kevin O'Kelly. Go ahead.
I hit the wrong button. Yeah, Kevin here. What do the animal trials look like there, Greg?
It's a regression, Kevin. Start off with usually like small animal trials, mouse trials, and those generally aren't used for like regulatory processes, but they would be. I think you're familiar with all of this. There's an Cook process when you get into animal trials where you need to show maybe the least amount of what damage or the least amount of suffering in your trial. So you want to start off. A lot of times before you ultimately get into trials that might involve like porcine trials, larger animal trials, they'll want to see that you've done some work with the smaller animals and that you're not introducing something that's really dangerous and painful. So that's the start, and then the next phase would be there's a standard set of trials for this kind of technology that involve porcine because they have similar characteristics to particularly human skin and wounds.
Thank you.
If there are no other questions, I want to thank everyone in attending and thank especially our speaker, Dr. Bertsch, for his time. And just as a quick reminder, our next presentation is November 14th with Dr. William Chen from BBS. So please remember to register so the presentation appears on your calendar. But yeah, thank you very much and have a good day, everyone.